we decided to move away from the blood smears and physical counting of white blood cells that has been done in previous years, and instead use flow cytometry for more efficient and accurate counts of white blood cells. In particular, we looked at ratios of lymphocytes to granulocytes and monocytes to determine the specific immune responses found in each treatment group. Surprisingly, we were able to extract a significant amount of blood from the individual zebrafish, which led to fairly successful flow cytometry data. Each week, the blood from 10 fish per group was pooled and stored in trisodium citrate as an anticoagulant, then taken to the Texas A&M flow cytometry cord laboratory for analysis. We simply used forward and side scatter analysis to determine the relative size and relative granularity of the cells in each sample. The first week, we didn't account for the fact that red blood cells would cause significant clutter in our data, which led to difficulty in seeing groups of white blood cells clearly. So in the weeks following, we applied a red blood cell lysis solution to the samples before running them through the flow cytometer. This gave us remarkably more clear results through which we were able to determine approximate ratios of lymphocytes to granulocytes and monocytes. Although, due to the fact that we did not use fluorescent antibody tags and only used forward and side scatter analysis, we've only been able to approximate the number of cells and the types of white blood cells in the samples. So what that means is that the groups that we've labeled as lymphocytes and labeled as granulocytes and monocytes may not in actuality be those types of white blood cells. However, what we found in our data was that the mucosal exposure group consistently had slightly higher ratios than the others, meaning that this exposure led to the greatest specific immune response in the fish.